I would like to go back to the technique. Is Ralph Weissleder on the line? Ralph?
Yes, I'm here.
Oh, very good to hear you. Sorry that you are not here. Thank you for sharing your precious time with us. We are a few minutes late, sorry, but thank you for your patience. We are very much looking forward to your presentation, and I think everybody here in the room knows Ralph Weissleder. He is coming from the imaging field. He worked in interventional, or still working in interventional radiology, and was a pioneer as far as molecular imaging is concerned. He trained an entire army of young German fellows who worked with him in Boston and then returned back to Europe. And he is developed in the last few years a very important and influential institute for system biology at the MGH in Boston, and he still keeps his contacts to the imaging field. And particularly for us, he was very important because he's on the advisory board of this building and helped us, especially in the early time, to get the right direction in the recruitment process for the research group here at the TranslaTUM. Thank you very much, Ralph, for this input. We will get you back in a few months. It was all interrupted by the corona situation that we couldn't invite you for an advisory board meeting. But today he will give us a talk about future life science laboratories, and as I know Ralph, it will be a very futuristic talk, and he will hopefully tell us a little bit about the thoughts in the Institute for System Biology at the MGH. Again, Ralph, thank you for joining us.
Thank you, Markus, for the kind introduction. You may just have jinxed this presentation. So my apologies for not being able to be in Munich today, but our institution still has an international travel ban, so none of us are allowed to leave and get reimbursed at the same time. So I also wanted to apologize for the corny title. It's a little corny, The Evolution of My Lab at MGH HMS. And it was due primarily to a slight misunderstanding of the session title. But the point I wanted to make is that there are many parallels between the center that I currently direct and the TranslaTUM straddling the basic sciences at a university and clinical practice in the trenches at a major general hospital. So what I'd like to do is give you a brief overview of how we have morphed from a pure imaging lab to a more molecular lab, and show some areas where these fields overlap and how we're striving to advance medicine and science in general. So let me start by giving you a short timeline. So my first lab at Mass General was called CMIR, or the Center for Molecular Imaging Research. This was one of the first molecular imaging research centers in the United States. It was created in 1993, so over 30 years ago, to develop and harness the extraordinary opportunities in molecular imaging at that time. So the lab at that time was primarily focused on pharmaceuticals. We developed targeted contrast agents for MR, iron oxide nanoparticles. We developed optical imaging, hybrid imaging, and focused on miniaturization. And so we did this very successfully for about 15 years. And then another extraordinary opportunity opened up at the end of the 2000s, when the institution here decided to build a new building like the TranslaTUM was built in Munich. And the institution decided to create interdisciplinary centers and And I was chosen as the chief to explore at that time physiologic genomics. And since no one knew what physiologic genomics was, it was renamed the Center for Systems Biology in 2009. And so what do we do? This is a map of natural language processing of our papers over the last couple of years. And so we work basically in three main groups. The red group, which is still related to imaging, blue, single cell analysis, biology and therapeutics, and green is inflammation biology or systems immunology. So 2 years ago, our center was renewed for another 10 years. With a primary focus on human systems biology or understanding how biology works in our patient populations. So while there is no specific mandate on the research, the principles that govern how our center operates is really the best athletes, thematic features, and the overall structure. So as in any large center, people grow up and they move elsewhere. So notable alumni who you will recognize, superstars in their own rights, are shown here. Mikhail Pitet, he was one of the first postdocs, rose all the way to full professor within the Harvard system, and just left this year and is now heading the Agora Cancer Center in Lausanne. He's also chair of immuno-oncology in Geneva. Phil Swirsky, another superstar, just became a director of a new cardiovascular research center in New York. Brad Bernstein was another one of our PIs. He is Mr. Epigenetics, if you wish, and so on. So currently in the center, we have 12 PIs at the junior mid-level. Matthias Narendorf, another German, is still around. Camilla Naxarova, another German, is still around. So currently we are about 40 faculty and about 200 people within the center. And so going back, this center was really a little bit of an experiment at Mass General to see how people from different backgrounds and different clinical specialties could work together. And they sure did. So over the years, we have had many high impact publications, and some of these have really opened up entire new field in biology and medicine and even in imaging. So this trend continues, and I think one of the underpinning reasons for that is that we bring people from basic sciences and from the clinical departments together so that they can basically think out of the box. So what does my personal lab do? So in 2021, we focus primarily on cancer, specifically pancreatic cancer, head and neck cancer. A main interest is in single cell analysis of these cancers. We use the commonly used toolboxes of single cell RNA-seq, database mining, cytometry, and so on, all with the idea of classifying and diagnosing disease. For example, can we detect cancers much earlier than is currently possible? To look at immune responses in cancers, to look at treatment efficacies, drug delivery, subtyping, molecular subtyping. But as you will have guessed by now, while this is great and it works extremely well to write papers, none of these toolboxes are routinely used in our patient populations for clinical decision makings. And the reasons are that the current single cell RNA-seq techniques, they're way too expensive and far too slow for clinical decision making. In other words, the single cell pipeline is really not industrialized at this point. The current techniques also require quite a bit of tissues, so often necessitating surgical resections or surgical tissue specimens. There's a slow rate of sample acquisition. There's no reimbursement for any of this here. So there is actually a disincentive of doing this on every single patient who walks in unless there are grants available. And there's also competition in the research community for these very precious samples. Because all of this, our lab is developing new technologies for single cell analysis and for single exosome analysis, and some of them are listed here. So in the next 20 minutes or so, I wanted to show you sort of two new technologies that we're currently developing to bring single cell analytical tools to all of our patients at the institution. So I started by saying that tumor tissue is essential in clinical decision-making, in addition to imaging, of course. And so this is done by two ways, either by using a core biopsy where one uses these guns to obtain tissue fragments. These tissue samples are then embedded. They're cut and stained. The turnaround time is usually around a week. And because we use guns up to about 16 gauge needles, complications do occur in a fraction of our patients. The alternative to all of this is to use a fine needle, so much smaller gauge needle that samples not tissue, but individual cells. So all of this can be done during the same day with many options, either image-guided, endoscopy, bronchoscopy, and so on. So the challenge with all of this, of course, is that with conventional cytopathology, one only looks at H&E stains, and what we want to do is multiplexing, deep molecular multiplexing. So in order to enable this in single cells and obtained by fine needle aspirates, we have focused in developing an entire pipeline where we harvest cells, develop new cycling chemistries, instrumentation, automated analysis to then report back to physicians in the hospital on what cells we're dealing with. And so our goal, our own set goal, is to obtain same-day results of anywhere between 10 and 20, 40 markers in cells, fully automated at very low cost, so that clinical decisions can be made within the same day. Now, this looks great on a slide. And what would be some of the clinical applications? So the context of use examples, there's really three major ones. As I insinuated, same-day diagnostics. This is highly desirable in care centers such as ours, general hospitals such as ours, to improve patient throughput. But it's even more important, I believe, in global health, where patients come, they want to be treated the same day, and then they go back to their village. And so we developed this for breast cancer detection and analysis in Botswana several years ago. And so within several hours, no, actually within less than 1 hour, we were able to make the diagnosis. The second use case is to interrogate entire cancer cell pathways. So in patients going onto clinical trials, and I would say up by now, at least 80% of all of our cancer patients are enrolled in one or other therapeutic clinical trials. So the question is always, do they have a specific target of interest? And then following drug treatment, how is this target affected, and how is the downstream signaling affected? So multiplexing in single cell allows one to do this. And the third context of use example, of course, is for immunoprofiling, where we're interested in the whole panoply of immune cells and their functional states in a given sample. So I'll show you, in the interest of time, of course, I don't have time to go into all of this here. I'll just show you one example. Let me go back one step and throw in one slide before I show you a clinical example of the cycling technique. So all of you are familiar with cycling techniques that are used on histological sections, embedded sections. They generally do not work in single cells with fine needle aspirates because they use invariably harsh conditions to unstain sections, paraffin-embedded sections. So they use hydrogen peroxides, other oxidants, toxic lights, and that just kills the cells. So in order to develop novel cycling we have developed over the years a number of different cycling techniques. Originally, and this was published 6 or 7 years ago in Science Translational Medicine, we called ABCD, and it basically had an antibody with a photocleavable linker to which was attached a DNA barcode. So this has been licensed to NanoString, and if you buy a NanoString kit to do protein analysis, this is the technology behind it. More recently, we developed another one because this was from NanoString. Even for us, it was too expensive. So we developed this scanned method that is slightly different. It still uses the DNA barcode, but it hybridizes fluorochromes to a DNA barcode. And just by using a melting buffer or different conditions, one could dissociate that fluorochrome and thus unstain sections. So this was used for a number of studies. Most recently, we're using this chemical technology. It's a bioorthogonal technology that uses fluorochromes directly attached through this bifunctional linker, to which, after staining, to which we can click a quencher, black hole quencher, and thus unstain or destain these cells. And then there's this technology here. It's an emulative technology. Apologies for that. I guess I don't have a slide here because it's not published yet, but this is coming out very shortly. And so this is even working in live cells. So we can now do multiplexed imaging in live cells at very, very low costs. So I'll show you one example here. And so one of the unmet needs, as was alluded to earlier today in head and neck squamous cell cancer, is this whole CPS scoring, which is essentially PD-L1 assessment in biopsy approvals to see if patients are eligible for anti-PD-1 treatment. As you know, it's not always feasible because it requires a tissue biopsy. Sometimes because of scheduling, this can delay treatment by weeks. It's not highly predictive. It's highly limited in scope because it only looks at PD-L1. And so our goal is to do much more deeper multiplexing and do this, importantly, longitudinally during treatment, since this is now with fine needle aspirates rather than with a core biopsy, we can do this. I would also point out that our surgical oncologists, they're doing the fine needle aspirations or biopsies in their office, and it usually takes less than 10 minutes to obtain these samples, 2 hours to stain them, and within half a day, the results are out. So I'll show you one example here. This is about to come out in Clinical Cancer Research. We started with 10 patients for validation studies. We're up to about 100 patients by now. And the only thing that I want to drive home is number one is that we get a fairly representative sample of these fine needle aspirates, a fairly representative sample of immune cells within these lesions. And we can do profiling. But importantly, I'm showing here one example of one patient who received anti-CD40 and anti-PD-1 and was sampled every 2 weeks, or every couple of days, 2 weeks, 5 weeks, 7 weeks, and so on. And we did complete immune cell profiling in these samples. So without going into detail, you can see that this is a fairly powerful technology because it allows you to look specifically what is the composition of the immune cell in this patient or in the samples. What happens, let's say, to specific macrophage subtypes, to specific CD8 subtypes. How does TCF1 change, for example, or interferon gamma or granzyme B and so on, or dendritic cell populations or IL-12. So fairly powerful technology. So the second example that I wanted to highlight, and again, this has unique intersections with the imaging community, and that is exosome analysis. You're all familiar with exosomes and other circulating extracellular vesicles. We know that they're shed from tumors, both into circulations, but also into lymphatic fluid. They're initially removed by subcapsular sinus macrophages in regional lymph nodes, but so once this system is overwhelmed, they will find their way into general circulation. They have unique characteristics. They're extremely abundant compared to circulating cancer cells, much, much more abundant. They're fairly stable because they lack a lot of the enzymes that usually degrade tumor cells. They're highly accessible because they're circulating. They contain proteins and nucleic acids from their parental cells, and thus reflect to a certain degree the cell of origin. So we have spent a lot of time in developing exosome analysis, particularly single exosome analysis. And so the one clinical use case that I want to show you here is for intraductal papillary mucinous neoplasms, or IPMNs. So this has become a bit of a problem case, at least in our patient populations. So it turns out that these cystic lesions of the pancreas, they're fairly common. So for example, in older populations, as shown here, it is estimated that up to about 1% of the general population will have some cystic lesions in their pancreas. And obviously, if we follow up this 1% of the population, it becomes extraordinarily expensive. We know that only about 20% will develop into pancreatic cancers. And so it's really become an issue of how does one manage these patients. And while there's certain criteria based on how fast do these cysts grow, do they have mural enhancing nodules and so on, none of the imaging tools are 100% diagnostic. So there's clearly a need of blood-based biomarkers to separate the benign from the more malignant subtypes, or the ones that are going to progress to cancer. So we thought that maybe exosome analysis is the way to go here. So what we did is, first of all, we created PDX cell lines from IPMNs. They didn't exist early on. And we compared basically every single protein that has ever been associated with pancreatic malignancy and asked the question, are they present in pancreatic cancer, in PDX cell lines, or in IPMN cell lines? And as you can see here, some of them are, some of them are not. And it's a long, long list. And from this list, we culled it down to something like 12 markers, and then went into the clinic and did a prospective clinical trial in 86 patients who were operated at Mass General. They were either healthy, benign patients, had low-grade IPMN, so these are the ones that do not progress to pancreatic cancers, or high-grade that will progress or had progressed. So I'm showing you these curves for all the different markers here. So for MUC1, one of the mucins, no overlap. For MUC2, no overlap. MUC4, no overlap. MUC6, no overlap, and so on. So all of these markers that had been identified in the literature, some of them as exosome markers, such as GPC1, for example, we found no difference between low and benign, except for one single marker, and that was MUC5AC. And so this MUC5AC, it turns out, is a highly predictive marker for invasive early cancers, pancreatic cancers, and high-grade IPMNs. And if we combine this serum analysis now with imaging, the ROC will go, this is CT MRI alone in our patient populations, together with the serum analysis, it will go to 0.99. So it has really ended sort of the conundrum of what to do with this patient population. So obviously the next question is, what can we do to identify the earliest stages of pancreatic cancers? So MUC5AC in that population is, of course, not 100% sensitive and specific. And so we were particularly interested in developing single exosome methods to look at mutated KRAS and mutated P53 in exosomes. When we started with this, the questions that arose are, how common are these mutated oncoproteins in EVs? Do they exist in our normal population, particularly in our aged population? And does multiplexing increase, i.e. measurement of both of these mutated proteins in a single EV, does that increase specificity? So you may ask, well, why don't I just go and sequence the plasma or identify circulating cancer cells, or why don't I simply look at the mRNA in EVs? So it turns out that mRNA, particularly full-length mRNA, is actually not that common. It is estimated that if one looks at 1 million EVs from a P53 mutated cancer cell lines, there's only one EV that will have a full copy of the mRNA. But the proteins are much more abundant, and hence the rationale to do protein analysis rather than mRNA analysis in EV populations. So what we, so the other thing that became very clear by analyzing the serum or plasma from hundreds of patients here at MGH was that the bulk analytical techniques were not able to pick up these mutated proteins. And if we look at the cell lines, it turns out that on average for different cell lines, that only 1 to 2% of EVs or exosomes of KRAS positive cell lines, only 1 to 2% of the EVs actually will have the protein marker. And because of this, because 98% will not have it, because of this, one needs to do single EV analysis to catch these 1 to 2%. The 1 or 2% that do have the mutated protein, they're very bright. So you can see this. TFP is a pan-EV marker here. And so you can clearly see the mutated KRAS protein. We can also identify this in patient plasma. So here it's about 10 microliters of plasma from a patient. We're looking at 1,618. This is just one snapshot here, EVs total. And we estimated that 1.2% of them were positive for KRAS G12D. So clearly an indicator of pancreatic cancer. We also figured out what the background is in normal populations and so on. And so, so currently we're doing a large clinical trial in applying this to anyone undergoing pancreatic surgery at our hospital. But the one point I wanted to close with is sort of give you an indication of how good a tool this will be now and in the future. So using mathematical modeling where we understand how big tumors are, how many EVs they produce, how fast they shed them in the circulation, and how these EVs will be cleared, we're able to do mathematical modeling. And so this is currently our sort of best thinking of where we are. So let's look with the current biomarkers that we have. And so this is the mutated KRAS, P53, MUC5AC, a number of other markers together. We are currently able to detect pancreatic cancers with reasonable sensitivity, and which are about 0.1 cubic centimeters in size. So this is a 5 by 5 by 5 millimeter tumor with about 80 or 90% sensitivity, just by exosome analysis alone. And so this is fairly spectacular. If we're able to add additional markers, and by additional markers, I mean mutated CDKs and whatever else there is out there, we will be able to left shift this curve. And the ultimate goal is to take this detection threshold down to somewhere here around the 0.01 cubic centimeter tumors, or 10 cubic millimeter tumors, which is a tumor of 2 by 2 by 2 millimeters. So the point with all of this is, if we want to do early cancer detection, not just of pancreatic cancer, but any cancer, one really needs these single EV analytical technologies. Without them, with bulk EV analytical methods, I see no way of achieving such high sensitivities. So our lab has developed a number of these different approaches. The one I just highlighted for IPMNs was this DEST method, where we do an immunocapture. We have also done direct imaging, and we have also developed immuno-based PCR methods for identifying or for analyzing single EVs. So I will stop here and leave you with a take-home message that technology development is necessary if we are to understand human biology, and if we want to develop better tools in the future. So I also think that there's life beyond imaging, and that the new molecular technologies will further the field of imaging, of molecular imaging and nuclear imaging. So I'm not worried at all about the future. And I sort of agree with our friend Albert Einstein, who once said, "I never think of the future, it comes soon enough." So thank you very much for your attention, and happy to answer any questions.
Thank you very much, Ralph, for this very exciting talk. And I think we share all this vision that technology is necessary to promote or to advance our field and to advance our, I think the key is the early diagnosis of disease. And we discussed this morning some of this stepwise approach, that you really need methods which allow you to screen a relatively large population at low cost to pick up the risk population, which then you can where you can intensify the search for localization of the tumor abnormality. Your use case is the pancreas and the ENT tumors, as I understand you. Where do you think which tumor entity is most suitable for such in vitro or single analysis approach?
Yeah, so the only reason why I presented on those two specific types is because we had done most works, and it is really dictated by local collaborators who are willing to share samples or let us come to the operating room. and work with us very closely. We do work as well in lung cancer, in breast cancer, in colorectal cancer, and so I think it's useful in pretty much any cancer that we have looked at. So I don't think it's cancer specific. I would say, with exception maybe of lymphoma, leukemias, where you can just do whole cell analysis, you don't need to do exosome analysis. But with respect to solid cancers, I think there is no particular one.
It's pancreas makes sense because many other diagnostic tools are not as successful as in mother. Sure. Okay, other questions? Are there any? Maybe another question which is important for us in the TranslaTUM. How do you actually organize this? You're an innovation hub, and how do you link to your clinical colleagues? Is this by postdocs or by fellows, or is this a more systematic, programmatic interaction between the clinical units and your Center of System Biology?
Yeah, very good question. So we do this by targeted recruitment. And so half of our faculty are clinicians. So half of the 12 people are actually clinicians who have a clinical practice, who work either in the ICU, who work in surgery, who work in pathology, who work in radiology. And the other half are basic scientists. And so this is enough of a footprint, at least, in the clinical realm. And many of our faculty, so for example, our surgeon who is a PI here, will sort of spread the gospel in the clinic that this is available and has brought in tons of additional collaborations. The same thing in the cardiovascular space. And so it's a recipe for success, I would say. The targeted recruitment, of a portion of the faculty who also do clinical work. Not a lot of clinical work, I would say, but at least let's say 1 day a week. 1 day a week in the OR or 2 days a week in the OR.
Yeah, this is still, I guess, a luxurious advantage that you have, the colleagues which can reduce clinical work to do such translational networking, if I want to put. Okay, other questions? Yeah? Takes a few seconds to move the microphone. Yeah.
So I don't know whether you can hear me. Thank you, a great talk. I was wondering, where do you go with this? So, you can detect very small cancers, but I guess this is not to screen the general population, right? So
that's correct. So for example, if we take the IPMN, so these are patients who walk into our emergency room and they get a CAT scan because they have belly pain, right? And then someone finds the cystic lesion, and then they end up in the cystic lesion clinic, and then the cystic lesion clinic doesn't know what to do with that particular patient. So it is not, it's not screening of the patient population, but it's for triaging to figure out what we need to do once these incidentalomas are detected.
Thank you.
Yeah.
Any further questions? Doesn't seem to be the case. Again, Ralph, thank you very much. And in a few hours, we're here in a beer garden on a rooftop. I hope you miss this. No, we would have enjoyed very much to also be with you at this very nice location, but maybe Boston will open up the travel ban and you can come at another occasion. And we will invite you definitely in the near future to advise us in the future of our TranslaTUM. Okay, thank you very much, and bye-bye.
Bye.
Now I would like to ask Ignasi Carrio to continue with the or start the moderation. Yeah.
Thank you very much. I'm very pleased to moderate the next session of this interesting and intense symposium that, in my view, it is run at least in part to honor Markus Schwaiger. I see many young people in the audience, which is wonderful for our field. Maybe some of you, because just a short time in the field, may not be fully aware of the importance and stature of Markus Schwaiger as a researcher himself, as a leader of a strong and powerful group, and as mentor. I believe it is now fair to recall that the strong group developed here in Munich by Markus Schwaiger has become, over the years, sort of the cradle of modern European nuclear medicine and molecular imaging, with plenty of people who started here, were raised here, now being heads of department, leaders of important labs, directors of departments, and so on. An amazing achievement and an amazing, I would say, legacy. In a way, it is important. In my view, it is impossible to understand the history of our field, nuclear medicine and molecular imaging, at a global scale. It is impossible to understand that without knowing who Markus Schwaiger has been and is, or is and has been. And having said so, I will continue with the session of this very intense program calling the next bright scientist to present on his data. Dieter Saur, please. Could you come? Oh, I'm sorry.
I'm already here.
Please, go ahead.
Thanks a lot for this very nice introduction, and I can just agree. It's really impressive being here. I would like to thank the organizers for organizing this, Markus Schwaiger, as well as Thorsten Gerken. I want to present what we have done the last 12 years. So we are since the beginning within this SFB, and we are very pleased about that. And our main focus is solid tumors. As you can see here, we are focusing mainly on the pancreas, but also work on liver and bile duct cancer, as well as in colorectal cancer. And these are the main killers we have in the clinic, except lung cancer. And especially these cancer entities here, they increase in their incidence, and so there's a clear medical need to tackle them. How can we do that? As you all know, it's a very difficult and complex process to develop novel therapies and to bring them into the clinic, which really make an impact. And I just highlighted here the different steps, and I think it's really impressive how this SFB impacted and still impacts on all these different points here up to the clinic, and showed already success in clinical trials. We are mainly working here in these areas, so we have a very strong focus in disease modeling. That's shown here. So we come up from human patients, look at their mutation profiles, at their tumor subtypes, develop model systems for these tumors, organoids 2D cultures, but also animal models, PDX models. Also sophisticated models such as transposon lines. Roland Rath is one of the pioneers here, CRISPR modifying, but also try to develop pig models for the disease, which is for imaging a very important model, in my opinion. And we have spread these models over the whole world in the last years, and are very happy that they impacted the field quite a bit. In addition, we are trying to develop technology that's done by Roland Ralph, for example, by functional screens. We contributed to this Nature with Cancer paper. We developed high-throughput pharmacological screens where we can profile tumors, see which subtypes are susceptible to specific therapies, develop combination therapies, and also use CRISPR and Cas to identify targets and also synthetic lethal interactions of genes. And in addition, we are trying to understand the biology of cancer by doing, for example, cell type specific in vivo proteomics here in Munich with the Max Planck Institute, but also doing a lot of the single cell analysis Ralph Weissleder already mentioned, which I think is really key, not only in the preclinical models, but also when we go then into the patients to understand these tumors in a much more comprehensive way, and I will show you some examples for that. So what we did as I said, in the last 15 years, maybe we worked a bit on prevention and early detection of GI cancers. As you might know, these flat adenomas are often missed during colonoscopy. Sometimes you have to stain them to see them better. This is another example here from our clinic. And what turned out to be the case is that the skills of the endoscopist, they impact a lot on cancer development of their patients. So if the adenoma detection rate is high, then the patients have nearly no risk of developing cancer, whereas if the adenoma detection rate of the individual endoscopist is low, then you have quite some risk in developing cancer after colonoscopy. And what we tried to develop was one mouse model for these flat adenomas. These are serrated tumors, and that was possible by using a BRAF mutation, and thereby we get these flat adenomas which are invasive and also progress to tumors, to invasive tumors and to metastasis. And what we then did together with Vasilis, I think we have to start the video, please. Molecular imaging techniques to see these flat adenomas, which we do not see. I hope the video will work. Maybe I have to just go on. Yeah, now it works. So macroscopically, it's not possible to see them, but by using molecular imaging probes developed by Ralph Weissleder, for example, that's possible. And we moved on with such approaches also in a more human setting, using APC mutant pigs, which developed these small adenomas here, as you can see, and we could also then do colonoscopy and colonoscopic imaging, and that's an example which we did together with Susanne, where we used an CCK2 receptor agonist, where we can, let's see if it works now, where we can detect also such small lesions. So that was in the beginning of the SFB, but of course it's also important to understand tumor biology and to develop novel therapies, and that I wanted to show you now with the next few slides and with some examples what we were working on. So one really intriguing finding with all these disease models was that we have a lot of tissue specificity. And that's summarized in this review, and it's shown by this data here very shortly that if you have a PI3K mutation in the intestine, then animals develop these serrated adenomas. And if we now delete PDK1, which is a downstream target of PI3K signaling, and if we delete this target in the pancreas, we do not get any tumors. So these animals survive. which shows that this is an important downstream target, and I will show you the pancreatic data later on. But if you do the same now in the intestine, these animals develop tumors much earlier, and they get a shift of their tumor, so which shows plasticity in the end, from these serrated adenomas to these mucinous adenocarcinomas. Another example for this tissue specificity is this bile duct cancer model, which we developed recently. By using Cre driver lines, we can activate in different tissues an oncogene. Here it's shown, it's the gallbladder, the common bile duct, the intestine, and the pancreas. So we have activation of an oncogene in 4 different tissues, but we get only then tumors in very distinct tissues. And here for KRAS, we see tumor development, of course, in the pancreas, but we do not see any tumor development in the bile duct. And then we looked into the molecular mechanisms, and we did forward genetic screens together with Roland, transposon screens, and we could identify signaling nodes that are important for tumor development in the pancreas versus the bile duct. And that's shown here a bit. So with PI3K, if we activate oncogenic PI3K, then we get bile duct cancer in nearly all of the cases. But with KRAS, we do not get it. We only get pancreatic cancer. And now if we inactivate P27, then we can shift, or then we can induce bile duct cancers also in the KRAS model. So P27 is the tumor suppressor that safeguards the bile duct towards oncogenic transformation. And I think that shows clearly that it's not possible then translated to the clinic to extrapolate findings from one tumor entity to another one, because the tumor suppressor nodes and the signaling requirements are completely different. So PI3K is an important pathway in the development of bile duct cancer, but also of pancreatic cancer. And we then ask the question, can we image PI3K activity? Because that inhibition of PI3K, the PI3K pathway, we found that this is an important therapeutic target. And first, we validated that PI3K is really activated in these human tumors. We identified a mouse model and mouse models which have high activation of these pathways. We also then found that PI3K signaling activity is linked to glucose uptake, so we can image that by FDG PET, and we can then, by blockade of PI3K, we can block glucose uptake in these tumors. And that's shown here then that we can image therapeutic efficacy in our preclinical models very nicely by doing FDG PET and thereby validate pathway inactivation. Another approach was to target epigenetic pathways, and that was one of the main achievements of Günther Schneider in the last years, that he developed very sophisticated combination therapies targeting epigenetics. And that's one example of his studies here, the combination of a BET inhibitor together with an HDAC inhibitor, which shows a high efficacy in treating patient-derived PDAC xenografts. How we can use these disease models to understand tumor biology better is shown here. So here we performed a multi-omics profiling of these tumors, and what we identified was that KRAS amplification and KRAS gene dosage, the increase of KRAS gene dosage has an important impact on tumor biology. So KRAS amplification mediates a more mesenchymal and more aggressive tumor subtype, which has a high metastatic potential. And we ask the next, does that also confer specific drug sensitivities and does it confer a very specific tumor microenvironment. And in order to analyze this tumor microenvironment, we perform single-cell RNA sequencing as well as multiplex histochemistry, immunohistochemistry, and we could find that these classical tumors, which are KRAS low, that they that are very differentiated with the highest tumor content, that they are driven mainly by CXCL5 and CXCL1, which recruits then myeloid-derived suppressor cells and thereby mediates immunosuppression. Whereas these very aggressive mesenchymal tumors, they mainly signal via CXCL12 and CSF1 and recruit a lot of macrophages, which then mediate immunosuppression. So immunosuppression is mediated by different pathways in different tumor subtypes, but the outcome is in the end the same. In order then and that's shown here, so these mesenchymal tumors, they are highly therapeutic resistant, so they do not respond to standard of care therapy. In order to develop now therapies for this aggressive subtype, we performed combinatorial drug screening with about more than 400 drugs and used specifically these mesenchymal tumors. And thereby we came up with a combination therapy of two clinically approved agents, trametinib and nintedanib. And this has a very nice synergistic impact on the tumors, as you can see here. So these are the mesenchymal tumors, and what we then, by profiling of the tumor microenvironment, what we observed was that we get an increased T-cell infiltration into these tumors, and that we can sensitize these tumors then to immune checkpoint blockade, as you can see here. And these tumors now respond as good as the classical subtype in the clinical setting by this combination therapy. So we are quite hopeful that this will also have an impact in patients in the clinic in the end. In order to identify the targets of our drug combination, because nintedanib is a multi-kinase inhibitor, we performed then together with Roland Rath genome-wide CRISPR screens, and as you can see, these are the targets of nintedanib that really have an impact on survival of the tumor cells, whereas other targets such as TGF-beta1 receptor confers resistance, which means we might also be able, by applying such screens, to identify drugs that even fit better with trametinib in combination than nintedanib. What is really nice by these genetic screens is that we can identify then the signaling nodes and the pathways, which are important for therapeutic response, as you can see here. And these are now the single cell data. So what we see is by applying this combination therapy here above is the mesenchymal subtype, we see a huge increase in CD8 cytotoxic effector T cells and in these mesenchymal tumors, whereas we do not see that in the epithelial tumors, which do not respond to checkpoint inhibition. In order to analyze the driving cytokines and the secretome of the tumors, we perform secretome analysis, as you can see here. And here again, we found that CXCL12 and CXCL16 is upregulated by the therapy, and thereby we can we have a mechanistic explanation why we get an increase in T cell infiltration as well as a decrease of these myeloid-derived suppressor cells and the M2-like macrophages. What Günther Schneider is working since many years together also with Uli Keller, who presented before, is on targeting MYC. And I wanted to show you a few examples how that works, and that there are subtypes of pancreatic cancer which respond to the therapy. There are different strategies to block MYC, as you can see here. MYC amplification, as Uli Keller already showed, is a marker of poor survival, so these are aggressive tumors. MYC expression is high in about 20% of pancreatic tumors, so we can thereby select these patients that have a MYC amplification. And then Günther developed synthetic lethal drug treatments and developed a proteasome targeting strategy which is highly efficient in these tumors. Another strategy is what he did together with Uli Keller, is to target SUMO. And that's a study which has been published last year, where blockade of SUMO is specifically efficient in MYC high tumors. So MYC can serve as a stratification marker in the end for such therapies. And that's in the end the concept then to by imaging stratify patients for such therapies. And one approach is to do this by annotating the MYC status, for example, by transferring PET imaging. So PET or in vivo imaging can serve here as a tool to stratify patients for therapy. That's an overview of the overall translational program we set up here at TranslaTUM together with Günther, Roland, and many other people that are shown here. So what we try to do is to get living cancer cells from patients as well as from preclinical models, organoids, cell lines, but also profile the cancer tissue and then characterize it and importantly functionalize it by CRISPR screens, high throughput drug screens, and so on, and then inform therapy. And one example for how that works is shown here. So that's work of Günther, where a patient before chemotherapy was biopsied, then an organoid line was derived, then the patient received FOLFIRINOX, that's the standard of care therapy for pancreatic cancer, and then post-chemotherapy, they generated again organoids from these patients, so we have before and after. And here is the timeline of the therapy of this patient. You can image them, of course, by PET. I was very, very interested in the data Johannes Notny showed today. I think maybe integrin imaging will be even better in the end. Then we need, of course, histology for diagnosis. These are the organoids. And then we can profile these tumors and see that there are quite strong changes and also metabolic changes in these tumors upon therapy. So there's a lot of plasticity, which is not driven by genetics, as you can see here. That's a sequencing profile of the two lines before and after therapy, and there are no major genomic changes on these tumors. But now these cell lines can now be used for high throughput drug screens again, and as you can see, we then could find some drugs which are more efficient now in the drug-treated tumors, which can then be used to treat these patients in an adjuvant setting, for example, after surgical resection. So with that, I want to finish my talk. I think, I hope I could show you that we try to do advanced tissue and genetic engineering, generate predictive models for which inform us for therapy stratification, but also for therapy selection. We benefited a lot from the imaging expertise in this SFB, and we try to implement novel tools. Ralph Weissleder already showed in his talk very impressively to better characterize these tumors and to really understand what's going on under therapy. We not only want to understand how a therapy works on the tumor cells, we also want to understand how a therapy works on the microenvironmental cells, how the immune cells are reprogrammed, which cells are recruited, and so on. With that, I want to finish. I would like to thank all the collaborators within the SFB, our teams of Günther and me. I would like to thank Günther. So he has been appointed a professor in Göttingen. He will start beginning of next month working there. So he will leave us, unfortunately. But it's, of course, also a great opportunity for him. But for me, it's very sad after 20 years of collaborations. And I would like to thank Markus Schwaiger for setting up this great SFB and for doing all of that. So thanks a lot, and I'm happy to take your questions.
That was very exciting. I like your concept of reverse translation very much.
Thanks.
We have time for one question.
Dieter, thank you very much for your talk. If you look back, or you're here with this institution for long, what are our bottlenecks to do the next step to bring all this, advances together? A little bit, I would like to connect the view of Ralph and your view. What would be the right approach for Klinikum Rechtsdienst for prolonging life in patients with pancreatic cancer?
I mean, I think we are quite on a good way because we have established quite a lot now and have set up these pipelines. It's extremely difficult to do all that, and it's also very cost intensive. So one thing I think we need to sustain this funding, which is very important. And then on the other hand, I think we need a better infrastructure for early clinical trials, more help with bringing therapies into these early phases. And with that, I think it would be already that would be quite a big step already forward. And I mean that's planned. And then we need more technology. I think that's the other important point, which came also out from Ralph Weissleder's talk. I think technology is the key enabler of novel findings, and but that's again expensive, and I think we have to try to work together and try to establish these things here and get the funding for that.
Yeah, but since I know now, familiar with both sides, the research side and the clinical side, aren't there some of these techniques ready to really go into the clinic? In my opinion, we have here this research environment and we have a relative traditional clinical management. How can we link this up in a way that we have this transfer of Nutain not only for clinical research but also for clinical routine? I mean, is single cell testing a clinical routine in anywhere, or is this all kind of the idea of the future?
I mean, at the moment there are many bottlenecks for these single cell technologies. It's extremely expensive. It's extremely laborious. Need a lot of expertise to do that, but I think it will be soon available. Sequencing is a good example. I mean, when 20 years ago when it started, it costed billions to sequence the human genome, and now in the end we can profile every patient, and I think that it will come. And I think profiling tumors before and after therapy will inform us a lot about responses, and if that can be taken from the blood, from the exosomes, maybe it would be great, but also from biopsies. I think that's clearly something which is, in my opinion, the future. With early diagnosis, I'm not so sure, as Mark already mentioned, screening patients or screening persons at risk will be very, very expensive and difficult. And then also there are then the false negative ones, which are, as the false positive ones, sorry, that where we do not find a tumor then, and then they get into this diagnostic Mühle, so to say. So I think for therapeutic monitoring, it will be very important in the future.
Thank you very much.
Thanks a lot.
Now the next speaker is Angela Krackhardt. Go ahead.
Dear colleagues, it's a great pleasure for me to be here today and to talk about the results of C10, a project which was invited to join for the second period of the SFB in 2013, and it's an honor to present data together with my co-PI, Calogero D'Alessandria. He will also talk a little bit, and we would like also to show other students actually working in the project. Two of them already left actually, but are still in the field of immunotherapy and in a context also imaging. And another one is just submitting, Christine Meyer, and we have very active students currently. I'm unfortunately not able to present all the work we are currently working on, but hopefully you will enjoy what we are presenting. So cancer immunotherapy is actually the subject of interest of my person. I'm a hematologist and medical oncologist, and as you all know, and as we heard a lot over the day today, is that we have increasing opportunities using cancer immunotherapy in a lot of disease entities, and the varieties actually of cancer immunotherapy and combinatory therapies are becoming broader and broader. However, there's a big lack of understanding actually of these immunotherapies, as you can see here. So the pharmacokinetics and also pharmacodynamics of these therapies are extremely complex because we are targeting a very complex system with these therapies. And we do see actually late responses, we do see mixed responses, we do see actually all kind of different responses which are not so, we are not so used actually from traditional chemotherapy regimens, and therefore we think that imaging is able actually to take over a very, very important part to get a better understanding of these therapies. For example, also in case of pseudoprogression, as you know. So we are talking today about the development of immunoimaging techniques to monitor tumor-reactive T cells in vivo. We have seen already very exciting data today about this subject. We are working on tracer development we will talk about. We saw already the data about the metabolic imaging of T cell activity in tumors and also epiluminescence imaging of T cells, and these were great opportunities actually we faced when we entered this SFB initiative. So in my group, we are especially interested in T cells, which are recognizing defined antigens. We are working especially on neoantigen-specific T cells, detection of neoantigen-specific T cell receptors, and we are transferring those T cell receptors by retroviral gene transfer or CRISPR-Cas9 to T cells, and then the idea is to expand those T cells and give them back. And there are a lot of clinical studies already undergoing using actually this technology and treating patients. And in the best case, we have then a PET-CT MRT monitoring actually available to understand what this therapy is doing in the patient and how the efficacy and also toxicity can be monitored. So the principle of immunoimaging, what we developed at the beginning was using a murine-ized actually T cell receptor recognizing a peptide derived from myeloperoxidase, and we used actually the murine-ized region in the part of the constant region for the imaging approach, as we used TCR-beta, murine TCR-beta actually antibody detecting those T cells. So we did our retroviral translation transduction, and then we established a mouse model which has, on the one hand, a leukemia, a solid leukemia sarcoma myeloid model with tumor cells which were expressing the right or the correct, actually, HLA binding motif, which was B7, and on the other one, one tumor which was expression B15, which should not have been detected. And on day 5, actually, we then looked if actually the T cells which we have applied to the mice were in the tumor and could be detected by our technology. So the principle of the experiment is shown here. So we injected the tumor cells into the mice. We then did a body irradiation. We injected our T-cell receptor transgenic central memory T cells, followed actually by the zirconium FAB IV injection with the anti-TCR-mu antibody, and then the PET CT imaging at day 5. And as you can see here, 48 hours after injection of those actually of the zirconium FAB fragment, there's a mistake. We have seen actually the tumor on the right position where we had the HLA matched tumors. In contrast, on the other side, we have not seen actually that signal. And the signal was in fact significant. So what we also learned from this model is that we see heterogeneity patterns of tumor-infiltrating T cells by using this tracking technology. So we did a lot of experiments and grouped actually the results into tumors which were really big and others which were engrafted less well and showed only low or small tumor sizes. And in fact, the T cells were certainly better capable in rejecting those tumors which were at the beginning actually at smaller size in contrast to those which were at bigger size. What we, however, also saw is that we were able to detect actually heterogeneous signal of the T cells in the tumor. This is the PET signal, the FDG PET signal, which we actually performed 1 day before, and this is the PET signal, zirconium PET signal of the tumors, and we saw this circular targeting of the tumor in case of these big-sized tumors. And when the tumors became smaller, actually, we saw a very central signal of the T cells. So by using actually this model, and maybe if we can translate that also to the clinics, we will be potentially able to see how is the dynamics actually of the T cell rejection in the tumor. And what you can see here is that we actually looked also if there's a correlation of T cell infiltration, and in fact we were able to see that at those parts where we saw a strong signal, actually we also saw a strong T cell infiltration in contrast to those actually areas where we had actually no signal and also no T cells. So we in my group actually, as said, are very interested in T cell receptor transgenic T cell transfer, adoptive transfer of T cells. However, there are other immunotherapies which are extremely exciting, and therefore it's certainly worth to work on a technology targeting actually all immunotherapies. And as you know already, there is CD8, for example, as an already clinically translated model to target T cells in the tumor. However, there could be others. To absolutely define, or for defining actually a T-cell monitoring tool, we do have to think about the target, we do have to think about the probe and the nucleotide selection. This all has to fit together in order to do a really successful clinical translation. So for pan T-cell marker, we were also we also wanted to include CD4 cells, for example, as also a complete picture, to provide a complete picture of T-cell infiltration. We were looking for different targets, and that were at the beginning CD2, CD3, and CD7. These are punt T-cell markers, and we were looking if they are expressed actually on our T-cells, as you can see here. And we were also interested if those markers are potentially expressed also on activated T-cells. As we heard already this morning, this will be a very interesting task to see if the T-cells are really on work. And that was given actually best for CD2, not for CD3 as a little bit expected, but also for CD7. So CD3, as you all know, can actually stimulate but also kill T cells. So therefore, this was more our positive control in the following experiments. And we then looked for full antibody as well as FAP fragments of CD2 as well as CD7 to see if this has a functional impact actually on our T cells, because these are our good guys, so we don't want to kill them, we don't want to harm them. And therefore, we were interested if there's any changes. First, we looked in vitro, and as you can see here, for those selected antibodies and FAP fragments, we did not see any change actually in the reactivity of the T cells. And also for the interferon response, this was quite similar when we used those constructs. When we then looked actually for specific stimulation, we saw a tiny difference, as you can see here, so a reduced elimination of target cells for those conditions where we had the right tumor cells and the right T cells for the CD2 FAP and full antibody, FAP fragment and full antibody, but not for CD7. When we then looked for the in vivo data, we saw a very nice signal at day 3 or 4 for the CD2, as well as the CD7 FAP2 actually imaging. However, when we then looked for a longer period, we saw that the tumors were well rejected for the condition where we used the anti-CD7 FAP2 fragment or the isotype, but not for the anti-CD2 actually FAP fragment, indicating that we have to be very careful when translating those tracers into the clinic. And now I would like to shortly give over to my co-PI. Tilo, please go ahead.
Thank you very much. And I just want to, at the beginning, thank a lot Professor Schwaiger for giving me the possibility to come into this very beautiful consortium together with Angela Krackhardt during the second finding period. So as Angela already showed, we were using till now FAB2, mainly produced by enzymatic digestion of full-size antibodies. But of course, we have to wait always, so at least 48 hours in order to get a high contrast image and to get rid out also of the circulating FAB2. And with the aim of a clinical translation of this imaging approach, we were thinking maybe on the possibility to use a little molecule, mainly antibody-derived molecule, that can fast penetrate into the tumor, reach the tumor, bind the tumor, and then but to do that, we need to use basically radioisotope that match also the half-life of this small molecule. Small molecule can for that small molecule we need usually to use fluoroethene or gallium-68. Fluoroethene, as is already known, so with his is characterized by having energy of 0.6 megaelectron volts, so and provide high resolution PET image due to also the linear energy transfer that is really favorable. And usually with its half-life also match perfectly with the pharmacokinetic of small molecule derived from antibody. Usually the radioprotonation of this small molecule, due to the fact that they are temperature sensitive, is really difficult, especially with the fluoroethene, because otherwise if you increase the temperature too much, then you destroy them. to do that, so we were trying now to approach this labeling with using the aluminum fluoride methodology. In this case, so what we need is a pentatendate ligand that is derived with because of the negatively charged oxygen donor groups that is present in the chemical structure, and which leaves only one binding site open for the aluminum for the fluoro-18. The standard approach till now has been the use of a note calculator, but the note calculator, due to this structure, needs to request to use high temperature of around 100 degrees and to force the aluminum fluoride complex within this ring, and you get always a radiochemical yield of 75% with a serum stability of 87%. In order to get rid out of this, the need of increase the temperature, we were studying these aminopiperidine-based chelators. So we synthesized in collaboration with Professor Lorenzo Tei in the University of Alessandria during his period here, when he was a Humboldt Fellow under the supervision of Professor Schwaiger. So synthesizing these 3 ligands, and then we characterized these 3 ligands, and we took, after many tests we have done, so for radiolabeling, this one, which provide us at the beginning a radiochemical yield of 55% at pH 5 and a room temperature. This is really important because at room temperature we are sure that we do not induce any kind of degradation of the molecule we want to use as a probe. The most important information is that the stability of this complex was over 90%, around 90% after 4 hours, which is definitely better than any other chelator for aluminum fluoride methodology that has been published till now. We tested also the stability in vivo, and as you can see also, what is really important, 2-hour post-injection, we are in the range of 1.63 to a little bit more than 2, which is definitely really good in comparison with the other published chelators. Now we are trying to functionalize this chelator in order to attach to little molecule or the little derived antibody-derived molecules, but we have seen that basically the TuREA affect negatively the radiolabeling. Therefore, we were now synthesizing a malamide derivative with a PEG in order to make this group very far or a little bit far from the coordination part. On the same time, we were also developing chelators for the gallium labeling, which can allow us to label also room temperature. And what we have done, we have developed of course, with the possibility that we can use gallium-68, we have developed a rigidified ADSTA-like ligand, which we published a few years ago, and which showed that provide a room temperature, a radiochemical yield of 80.7%, and with a high stability. So now one of our PhD students is working on the functionalization of this chelator for gallium labeling, which could be an alternative. Why we are working also on this chelator, simply because we have found that this chelator is able to complex very well not only gallium-68 but also scandium-44, which is with his 4 hours half-life. So it's really interesting as a isotope to use a small molecule, especially protein temperature sensitive molecule, and also is suitable to complex indium. And this has been already published by some other colleagues already in Angewandte Chemie and recently in European Journal of Nuclear Medicine, in particular because they were able to complexes of scandium-44 and radiolabel a new ligand for PSMA. So it could be something for the future. And now I would like to give again the word to Angela.
Just for finalizing, I mean, we have seen these data already. I think these technologies, and it's so important to see all this chemistry behind and also the physicists behind to see what we can do if we do have, for example, imaging for pH, and see what is the acid environment in the tumor doing on the T cells, and how can we improve our therapies actually to let the T cells go into this acidic milieu, which we have observed actually in our models. And also the 3D reconstitutions actually, which can provide more information to us how the dynamics of the rejection of the tumor can be improved by selected therapies. And by that, I just wanted to show some publications coming out of the last period. I would like to say that I'm extremely grateful for the opportunity to work in this consortium. Cross-disciplinarity is, I think, one important sign of this consortium, and these two guys are currently working on an extremely interesting project we will demonstrate next time. And finally, I would like to thank especially Markus Schwaiger for this great initiative and all these wonderful people we work together with. Thank you very much for your attention.
Thank you very much, very impressive work. When a patient is considered candidate for a CAR T therapy and enters the program, how do you foresee the potential role of an imaging technique to demonstrate either T-cell infiltration or rejection? How could that be put into the program?
Absolutely, very important question. So one thing, I mean, for the CAR T-cell patient, I would consider a patient who has a very aggressive disease where we give the T-cells, the CAR T-cells, and then we do not have a lot of time actually to wait if he or she responds. And that's a very important time point where we can see if the T-cells survive, do they engraft, do they go into the tumor? This is, I think, a very, very important application. And in solid tumors where we actually may want to predict a response to immunotherapy, there I think a pan-T-cell imaging would help actually to see are the good guys in the tumor, can we do stimulate them by any immunotherapy we plan to do.
Thank you. Any other question before the coffee break? If not, thank you very much. How much time do we have for the coffee break? 10? 15 minutes. So rush for the coffee. Let's go on for this last part of the afternoon session. And now Christina Spitzweg will take the lead.
Yeah, thank you very much. It's really my pleasure to present our data together with my two PhD students. I thought it's refreshing if also the young people come to the front and talk a little bit about their ongoing work. It's my pleasure to present our data on the optimization and individualization of the sodium iodide SymPort NIS-based gene therapy in glioblastoma and also other tumors based on characterization of tumor heterogeneity and tumor micromilieus. Probably almost all of you know the sodium iodide SymPort pretty well by now after 12 years of SFB because it's been the major focus of our work, of our group. The sodium iodide SymPort NIS is clearly one of the oldest targets and one of the most successful targets for molecular imaging and also for targeted radionuclide therapy. It's the molecular basis for the diagnostic and therapeutic application of radioiodine that is celebrating its 80th anniversary this year. And cloning of NIS actually is also celebrating an anniversary. It's the 25th anniversary this year of cloning of the sodium iodide SymPort. And that has opened really one of the most exciting fields in molecular thyroidology, with the characterization of NIS as a theranostic, as one of the most promising theranostic genes, and the development of a new gene therapy concept, gene therapy concept based on image-guided selective transfer of the sodium iodide symporter to non-thyroidal tumor cells, and followed by therapeutic application of radioiodine or alternative radionuclides like a statin or rhenium. And after I have started this work on the NIS gene therapy concept many years ago during my postdoc fellowship at the Mayo Clinic, the SFB, the 12 years of the SFB really provided us with the platform, with the tools, with the scientific environment that we needed to move this NIS gene therapy field from local NIS gene delivery to new promising applications for disseminated disease, which of course is our goal. In particular, concentrating on polymers, non-viral polymers, and genetically engineered mesenchymal stem cells as highly effective, highly selective, and flexible tools for further translation of the NIS gene therapy concept in the clinic. And in the recent in the last funding period, we have also focused on combination.
Welcome back. I would like to go on with the multimodal imaging. For the setup, we used patient-derived xenografts model from the B2 project, Professor Klaas. After the metabolic imaging with MRI took place using hyperpolarized 13C substances, hypoxia PET imaging was performed, followed by a single MSC application using the hypoxia responsiveness MSCs, and the readout took place using iodide 124 PET imaging. For the hypoxia imaging, FMISO was used as a well-known PET tracer, indicating pronounced hypoxia within this tumor model, which was expected and was further confirmed by injection of pimonidazole, which can be detected ex vivo by immunohistochemistry. The immunohistochemistry and the analysis of the immunohistochemistry took place in close collaboration with the Institute of Pathology. After the hypoxia imaging After the hypoxia imaging took place, a single MSC application of the HIFNIS MSCs was applied, and the radioiodine PET imaging showed a tumoral accumulation, which could be blocked upon treatment with the NIS-specific inhibitor perchlorate. This was the tumors were further analyzed ex vivo, showing MSC recruitment per se by CMFDA labeling of the MSCs, and this expression could further confirm the MSC-mediated radioiodine uptake in the tumors. We could show a very strong correlation between hypoxia and the hypoxia-responsive MSC-mediated iodine biodistribution. On one hand, in vivo by PET imaging, and also ex vivo by immunofluorescence staining. All in all, the study shows very promising preliminary data for an hypoxia imaging-guided MSC-mediated NIS gene therapy concept by the use of hypoxia selective NIS-expressing MSCs. And now I would like to come to an end and with the MSC-mediated approach, and would like to hand over to Rebecca for the polyplex-based approach.
So to apply the NIS-mediated radioiodide treatment of glioblastoma, in this project we worked with polymers. Polymers are inspired by viral biology, and they are designed for dynamic, potent, and targeted nucleic acid delivery. So in close collaboration with Professor Wagner's lab from the pharmacy department of the LMU, we established polyplexes based on sequence-defined cationic lipo-oligo that encase or complex the NISP and DNA by electrostatic interactions. On the surface of the core, there are acido groups to functionalize in the second step the polyplex by adding ligands containing DBCO for copper-free click reaction. Here you can see the structure of the lipo-oligomer, and we work with those ligands. So the DBCO group for the click reaction, as I mentioned, a PEG24 domain as a shielding domain to lower the surface charge, and the G11 as a targeting domain. G11 is an allosteric and specific ligand for the epidemic growth factor receptor that is a histopathological hallmark in glioblastoma, and we compared mono-DBCO agents to bis-DBCO agents. We characterized the polyplexes regarding their size and surface charge. They are between 120 and 140 nanometers for optimal biodistribution and pharmacokinetics, and the surface charge is lower than 20 millivolts to reduce the risk of aggregation with biomacromolecules or self-aggregation to provide a longer blood circulation. In transfection studies in vitro, we could show EGFR-dependent transfection efficiency by the transfection of cell lines with different EGFR receptor expression levels. We could show the advantageous effect of using G11 by transfecting the cells with a non-targeted polyplex as compared to the G11 targeted polyplexes. And by using the inhibitor perchlorate, we could show that the iodide uptake was NIS-mediated. For the in vivo studies, we established in our lab autotopic U87 glioblastoma xenograft mouse model. Here you can see the picture of a mouse brain containing the U87 tumor with the H&E staining. With the pathology, we stained the EGF receptor expression level in the U87 sections, and this brownish staining, you can see that we had a positive receptor expression. And in the PET imaging, we could show the advantageous effect of G11 polyplexes as well. So all three groups which received a GE11 polyplex showed a strong signal in the glioblastoma in the brain area, as compared to the non-targeted polyplexes. They have just a signal slightly above background levels. And by using polyplexes containing loop DNA instead of NIS DNA, we could show the NIS specificity. In the quantitative analysis of the tumor iodide uptake, we can see that the mono-DBCO G11 polyplex is the best performer. It's a significant higher tumor iodide uptake as compared to the non-targeted polyplex. We compared 48 hours between polyplex injection and iodide injection to 24 hours, and it worked better than the 24 hours. And the green line is a bis-DBCO agent, and we see that the mono-DBCO agent worked better than the bis-DBCO. So for the dosimetric calculations for iodide 131, we revealed a doses of 58 milligram per MBQ for the mono-DBCO G11 48 hours in between, as compared to the bis-DBCO G11 with 35 milligram per MBQ, or the negative control with a non-targeted polyplex with just 8.2 milligram per MBQ. We confirmed the positive NIX expression in the glioblastoma sections. So all three groups that received G11 polyplexes showed NIS-positive cells. The 48 hours show more NIS-positive cells than the 24 hours, and all other control groups do not show any positive NIS expression. We also stained the liver, the lung, the kidney, and the spleen as control organs, and here we don't have any NIS-positive cells either. So for the therapy study, we applied the mono-DBCO G11 polyplex. In 48 hours, we applied the iodide 131 and repeated this cycle for 3 times. We observed the tumor growth by performing MRI and the survival length, and the results from the therapy matched closely to the results from the imaging. We have a significant delay in the growth of the tumor in the therapy group as compared to the control groups, and we have a mild delay in the group that received the non-targeted polyplexes. And remember, they had a signal slightly above background, and here just a mild delay. And the outcome of the tumor growth behavior matched closely with the survival length, or is mirrored in the survival length, so that we have a significant extension of the survival in the therapy group as compared to all 3 control groups. Ex vivo, we stained the therapy tumors regarding their cell proliferation and their blood vessel density. And we can see that the therapy group has a trend towards lower cell proliferation shown by a Ki67 staining, and we have a significant lower blood vessel density shown by a CD31 staining, and this shows a long-term anti-angiogenic effect of iodide 131. As a next step, we want to target the blood-brain barrier. The blood-brain barrier is a restrictive barrier that insulates the neuronal system to provide an optimal function of the neuronal system, and the receptor-mediated transcytosis is a well-known mechanism to transport large biomolecules across the blood-brain barrier, and we focus on the transferrin receptor. So we want to establish a polyplex, a dual-target polyplex that has two targeting domains. First, the transferrin receptor to target the blood-brain barrier, and in a second step, to target the tumor by G11. We already characterized the dual-targeted polyplexes in vitro, and with Professor Wagner's lab, we want to establish a transfer model for blood-brain barrier targeting in vitro, so that we have a BBB monolayer, and in the acceptor chamber, the glioblastoma cells. So Caroline and I want to say thank you to Christine Spitzweg for supervising our projects and to our lab. Then we are thankful to Peter Nelson as an expert of the MSCs, and to Ernst Wagner as an expert of the polyplexes. Thank you for Theoman for all the synthesis of the polyplexes. We are thankful to the Department of Nuclear Medicine from the LMU and from the Technical University. So thank you to Professor Schwaiger and Professor Weber. We want to say thank you to Franz Silling and Martin Grasey for the collaboration within the hypoxia project. Thank you to the Bille Reda, Markus Mittelhäuser, Hannes Rubinski, and Sandra Sunil for the help in performing all the images. Thank you to the pathology for preparing the tissues, the staining, and analysis. Thank you to Gabriele Molotov and her team for the help and support in the radiation experiments. Thank you to Volker Morath for the support in the tetrafluoroborate. And thank you to Professor Glass and Roland Kehlin for providing the glioblastoma cells, and to all of you for your attention.
Thank you, Rebecca. And I would also like to take the opportunity, just a few more seconds, I would also like to take the opportunity to thank all the collaborators within this outstanding network that Professor Schwaiger has created. Many, many thanks to you, Professor Schwaiger, for creating this. This really has become my scientific family, and you are clearly the father of it. So it's been a privilege to be part of it, and I really hope that we can continue this dynamics for the future. Thank you so much. Thank you.
Thank you very much. That was a very nice presentation, despite the technical difficulties. Is there any final question for this group? Or if not, thank you very much. We will continue to the next presentation that will be given by Rickmer, Braren, and Irina Heid.
Okay, so welcome everybody. I will introduce the project, and then Irina Heid, my long-term collaborator, postdoc in my lab, and now becoming, I guess, her own lab boss very soon. She will present on the preclinical part, and I will introduce the clinical developments within this project. So the title of our project is Development of Prognostic Biomarkers, and we focused on two biomarkers, one which is primarily a structural marker, diffusion-weighted imaging, and the other one being FDG as a metabolic marker. And this we further expanded on then during the project in collaboration with Franz Schilling. You heard a lot about him and his work, and this is going to focus then on pyruvate and lactate imaging hyperpolarized at the 7 Tesla MRS. And okay, with this I hand over.
Good evening. Thank you very much for the opportunity to speak on this symposium, and I also thank everybody who helped me and believed in me to do this project. So, as Dieter Saur pointed out very nicely in his talk, molecular subtyping and subtyping at all plays a huge role in pancreatic cancer. And why? Because it has different subtypes have different survival and also different reaction to the therapies. Now our work is about predicting subtype before you do any intervention with the patient, predicting subtype but not by non-invasive imaging. To do so, we needed to advance our previously established very heterogeneous model of unknown subtype to the transplanted models of very well characterized and known subtypes. We established at least 2 epithelial and mesenchymal subtypes in the mouse as pancreatic autotropic allografts, and also we established the subcutaneous xenograft of human 2 cell lines in order to really detect different subtypes. Now, another huge advantage within the third period was the transition from the 1.5 clinical Tesla scanner, where we did our initial studies on tumor cellularity, to the 7 Tesla animal preclinical scanner with the help of Franz Schilling and his group, where we established and looked at very highly resolved anatomical imaging, but also highly resolved diffusion-weighted imaging that detects water movement within the tumor and gives clues about tumor composition. We confirmed previously established grouping of the tumors into the 3 by the tumor cellularity, the most cellular tumor having the lowest ADC. We confirmed that this also works on 7 Tesla and divided tumors now in 2 groups just to do it more easily. 40%, up to 40% tumor cell lines within the tumor are located here, and more than 40% tumor cell lines within the tumor are separated very nicely by this method. Oops, I'm sorry. Go away. Now, returning to the subtypes. Meanwhile, while we'll be establishing the methods, it has been published that the molecular subtypes based on transcriptome, on the biopsy of the cell, also can be translated into morphological subtypes. In this case, gland-forming and non-gland-forming. Now, this is something that also been proposed to be even better, maybe, than the classical and basal or quasi-mesenchymal subtype in terms of separation of survival of the patients. And we can use this because ductal structure contain more water than the solid tumors or tumors without ductal structures, and that's why we used first of all DWI, diffusion-weighted imaging, on our 2 clearly clarified models of epithelial and mesenchymal, and we use different parameters, less p-values that are more related to the clinical scanner. Then we explored the influence of higher p-value with this method. Within this method, we analyze cortosis and fast diffusion fraction. Unfortunately, despite apparent significance, there is major overlap between the subgroups. So we conclude that this method is insufficient to define subtypes within the tumors. And the same was unfortunately also true for the glandular and non-glandular tumors. They were also not separable by diffusion-weighted imaging. However, we had a second approach in mind, and that was the metabolism of the tumors. Classical tumors have been assumed to be non-glucolytic or less glucolytic, and the QM tumors were assumed to be highly glucolytic. We looked at it via FDG PET and seen that indeed uptake of glucose is a lot higher in the mesenchymal tumors than within the epithelial tumors. And also you can see here on this separation there is no overlap, and the subtypes are really clearly separated from one each other via FDG PET. And then we also established and used spectroscopic imaging of hyperpolarized pyruvate that is converted into lactate and vice versa. And both of the methods reveal the same result. Mesenchymal tumors have high glycolytic activity. They produce a lot more lactate and convert and also take up lactate and convert this into the pyruvate. But this was spectroscopy, it was a spectrum, it was not imaging, and we want to image, we want to see the different tumors and see the differences within the tumors. That's why we advance this imaging by chemical shift imaging, and as you can see here, tumors are visible on this imaging, and they also have different signal. Mesenchymal tumor, again, produce a lot more lactate than the epithelial, and using this model, because it's very well characterized and very clear, we also established immunohistological markers for our model of unknown subtype, and this is the monocarboxylateral spotter MCT4 that has been proposed to be highly expressed in glycolytic tumors. And here we see also on the discodification very clear separation between the epithelial and mesenchymal tumors. Now in the second step we use our highly complex and heterogeneous model. We also perform chemical shift imaging through the abdomen. We see here different tumors that derive different signal. And if we look through the whole population, we observed that these tumors marked in red within the cellularity group, because it's very important to divide this highly heterogeneous PDAC into two groups of similar similarity. Otherwise, you will have a tumor that produces a lot of lactate just because there are a lot many more cells than in the tumor that where is a very low cellularity. And oops, I'm sorry. Looking at this, we see that a highly lactate-producing tumor marked here in red within both groups appear to be quasi-mesenchymal of their appearance, but also they are highly expressed Vimentin, a mesenchymal biomarker, and also MCT4 is established. And those tumors with low signal in the hyperpolarized MRSI, they appear to be rather epithelial and don't have high expression of MCT4 and Vimentin. With this, we conclude that we can reliably stratify endogenous murine pancreatic cancer into the subtypes. And now, since we established with the help of Roy Schilling and Jason Skinner a 3D metabolic imaging, we can look at the whole mouse and their conversion of the pyruvate to lactate. We also advanced our structural imaging platform by adding T2 and T1 mapping as a quantitative MRI parameters. And we've also evolved a lot our post-processing program, where we derive cell lines from a really known region marked with the pathological ink. And these cell lines then will be correlated with the histology and stained for the certain biomarker, as well as we will do laser microdissection and derive molecular RNA-seq samples to analyze what transcriptional subtype also is underlying and correlated with this imaging. With that, yes, and we started to do therapeutic trials with these mice, and for us it's important to use the standard of care therapy to see how the tumors change under the standard of care chemotherapy, and to be able to predict what therapy is working or what not. Now you see here that the non-treated mice mostly stay normal, and within the treated group a lot of is ongoing, and we will be analyzing this within the next months and maybe years. Thank you very much for your attention. With this I will give over to my colleague Rickmer.
Okay, thank you very much. I think actually one point, maybe, I don't know if everybody got that in this slide, which I think is really a very important finding that I want to reiterate, because I think this is something that we may be able to translate really into a clinical scenario. So this group of tumors here has very low cellularity, and we can identify these tumors based on this high ADC value. And this group here has very high cellularity, and it's identified by a quite clear cutoff, again, by the ADC value. And so when you then start interpreting these sort of high within group signals that we find. This is something I think that we would really like to translate and test then on the PET-MR setting, where we can first group tumors based on tumor cellularity and then interpret maybe the glucose signal that we see based on the tumor type, which has high cellularity or low cellularity. Anyway, so translation into the clinical scenario is quite hard, difficult, and I guess Irina is drilling from a very preclinical side, and I'm coming from a very clinical side. And here I just would like to show 3 projects that we did in close collaboration with Dimitrios Karampinos, also PI within the SFB, who's working on body MRI, amongst others. Increasing resolution is really an important issue in diffusion-weighted imaging, and we managed to push this really quite far to get robust diffusion-weighted imaging in the pancreas with a very high resolution of 2.5 by 2.5 millimeters in plane and just 3 millimeter plane slice thickness. So this comes at some cost in MRI, like for instance noise bands that we generate due to these reduced fields and that increase with the reduced field, which then can generate artificially low ADC values, which is of course detrimental to the exact quantification of tumor cellularity, which we do based on ADC. And so we applied post-processing in these to reduce these noise bands and get more reliable ADC values. Or, for example, like the computing of high B values, so measuring diffusion-weighted measurements come at high cost in terms of scan time. And this is an image acquired, not acquired, but computed from acquired B values, which are measured up to B600. This is a computed B1000 image, and what you can see is that in this IPMM lesion, for example, there's a nodular structure, and this nodular structure nicely becomes demarked in this computed image because the T2 shine-through effect is reduced or abolished. So this is work related to really clinically applying diffusion-weighted imaging and translating these preclinical findings into the clinics. Magnetic resonance imaging, of course, has multiple limitations. It's expensive, it's not widely available, and radiologists in private practice oftentimes don't manage to generate such nice images because they don't have the same technical staff. Nevertheless, we wanted to translate these findings from the preclinic into the clinic, and so we did a study on spectral CT imaging and tried to do the same correlation between surgical specimen cellularity areas identified and the corresponding in vivo imaging of the pancreatic tumors. And we could really nicely separate these different cellularity levels based on Hounsfield units. These were normalized, of course, to the large vascular structures and could nicely separate between cellularity levels. And this had been previously shown that these, in a work by Irina, that these cellularity levels are really predictive of survival in a retrospective analysis of pancreatic cancer patients that were resected. Okay, so this is a very different work package. We also proposed that we would curate data, retrospective cohort of pancreatic cancer patients from our hospital. We expanded this, and I just want to report on a few studies that we ran retrospectively based on this imaging cohorts. We expanded it and were lucky enough to have several collaborators, Diane Simeon from MRU, Centers for Cancer Patient Files, and also several university hospitals. This is a study that aimed at identifying pancreatic cancer, early stage pancreatic cancer, based on CT imaging and machine learning. And what we did was we collected a cohort of patients that had early stage cancer or autoimmune pancreatitis. And a subset of these patients was actually resected because it was thought that they had developed a cancer. And so this was our validation cohort, and we could train a model to separate these nicely with a very good ROC AUC of 0.9, which is a way to, I guess, to prevent unnecessary surgery in such a patient cohort. Another application, so we could show you, or Irina was telling us that she could not differentiate subtypes based on ADC imaging. In our retrospective cohort, we applied machine learning to differentiate, at least based on the median survival of these patients between above and below median survival. Again, we could train a model that was externally validated on an independent cohort with a very good sensitivity, specificity, and again, ROC AUC of 0.9 to differentiate based on the preoperative imaging into patients that would survive above or below median with the same treatment regimen. We could also use CT data of this retrospective cohort to train a model to differentiate molecular subtypes. Molecular subtypes were confirmed by a histopathological marker, which is not 100%, but it's doable. We could stain these retrospectively for HNF1A and keratin 81 in collaboration with Katja Schwaiger from the Z, or pathology department and Z project, and could train this model to really nicely differentiate between these two subtypes, the QM and the non-QM. And we could even apply then this model to a cohort that could not be stained or had a staining that was not fully comprehensive in terms of this differentiation for technical reasons, I guess. And if you apply this model then to this unknown cohort, you clearly get two survival curves which correspond to the QM and the non-QM survival curve. So this could be or indicate one application of this preoperative imaging, especially in tumors that are very heterogeneous, where we could at least suggest that a tumor belongs to a certain molecular subtypes and should be treated accordingly. Current work is aiming at the integration of these imaging features into more complex models where we integrated, and this is just showing hazard ratios of imaging parameters or image-derived parameters in comparison to known parameters like tumor T or N staging or certain mutational status like P53. So they have some impact, and currently, we're moving this into an end-to-end modeling where imaging data together with metadata is integrated to generate image-derived reconstructions, for example, to classify based on vascular contact in which treatment scheme a particular patient should go based on these arterial contacts, and then to predict a specific or individual patient survival. So with this last study, I would like to conclude. And again, this is, I think, a good example how far this way is then into the translation. This is a PET-MR study that we started several years ago, actually, and managed to recruit 20 patients within. And the idea was to to image patients with PET-MR, FDG PET-MR before and 2 weeks after therapy onset. Patients receiving FOLFIRINOX combination treatment or gemcitabine Abraxane were included in this study. And this is the pre-post imaging where you see the anatomical imaging after 2 weeks shows no difference, but the FDG drops and the ADC increases in this patient that after 3 months shows a nice response here. And when you look at the numbers, there's no clear winner, I guess, but two FDG-derived metabolic tumor volume and total lesion glycolysis, and one MR-derived, the ADC mean value, came out as the most predictive markers for this response after 3 months. And this may be a good way, especially in diseases like pancreatic cancer, where survival times are short and options are established by now to then switch regimens based on such early response evaluation. So I'd like to summarize. So we've worked a lot in the last funding period on pancreatic cancer, or started working on pancreatic cancer, and did some preclinical testing of novel imaging technologies, established this imaging platform, generated a large retrospective cohort, which we now use to develop such models that we can test prospectively in the future. With this, I'd like to thank you all for your attention, especially again, Markus Schwaiger, for establishing this SFB, having me in there for several years, and we're happy to take your questions.
Thank you very much. Very interesting data. Is there any question for this group? Yes, please.
Thanks for this very nice presentation. I have a question for Irina. You showed nicely that by combining PET and hyperpolarized MR, you can basically improve first and last steps of diagnosis. Is there actually an additional benefit by looking at both steps for the subtractive differentiation? Because you could imagine by only having both steps, you would be able to look at refluxes and into the phosphate pathway. Does that play a role?
Thank you, Franz. It's a very interesting question, but as of for now, we weren't able to do both modalities on the same animals in the endogenous model. We have a subset of animals that receive PET and subset of animals that receive hyperpolarized MRI. And hyperpolarized MRI differentiates better than PET different tumors. And we have a subset of these transplanted models that really correlate very well, like R is 0.9 between PET and MRI. So I guess if the subtype really clear, then it derives kind of similar information. But if the subtype is unclear and maybe mixed, as it is in our genetically engineered models, then I could imagine that they complement each other, but we haven't been able to analyze this.
Thank you very much. For the sake of time, we have to move to the last presentation that will be given by Dr. Biedermann.
So I'm from the dermatology department and in the group of Tilo Biedermann, and we joined the SFB. We were grateful to Professor Schwaiger that we could come here when we moved from Tübingen to Munich, and our focus is more on the biology side, and I would like to present 2 projects about melanoma, targeting melanoma and associated immune cells. So first one will be about mast cells and target them to induce immune defense. And so when the treatment of patients with metastatic melanoma was completely revolutionized by immunotherapies and leading, as we heard this morning, to a sustained T-cell response. But these patients also develop immune-related adverse events, and in a project we did together with the dermatology in Tübingen, we looked for the best therapy response, and as you can see here, that those people that develop colitis have the best response compared to other adverse immune-related events. So colitis leads to an increased permeability of the intestine, so we allowing that sorry, I'm completely making it possible that a systemic exposure of microbial components. And so we asked whether we could identify biomarkers in the serum of patients and compared those with and without colitis, what is shown here, and identified an LPS signature. So that is a component of bacterial cell wall. And we hypothesized that this could contribute to therapy efficacy. And looking for potential target cells, we thought of primary melanoma where spontaneous regression mediated by T cells does occur, and indeed we identified a high accumulation of mast cells, here shown by tryptase staining, and here the quantification of this. And when we then generated a Z-score of mast cell signature genes, we applied this to the database of the TCGA from the NIH, then indeed we found that there is a significant better survival in patients that show a high mast cell gene expression in human melanoma. So moving then to preclinical model, we used a setup where we then used LPS as treatment, and also here we could find an accumulation of mast cells showing here. And going then in mouse models that are deficient of mast cells, we found that indeed the LPS treatment in wild-type mice initiated an effective anti-tumor immune response, while in those mice that lack mast cells, this was not the case. And we have the possibility to repopulate the skin of mast cell deficient mice with bone marrow derived mast cells, showing here with another mast cell deficient mouse model. And what we could show is that indeed only in the presence of mast cells we do have tumor control due to LPS activation, but not when mast cells are lacking. And without going into details, we screened for a while for the underlying mediator and could identify the chemokine CXCL10, and what is shown here that when we transplant mast cells deficient in CXCL10, then indeed the rescue of tumor control got lost again. And we could also show that this was mediated by recruitment of T cells. So CXCL10 is known to be chemoattractant, and we found that this leads, after selectively activating mast cells by LPS, leads to a recruitment of effector T cells due to the mast cell-derived CXCL10. And going back to melanoma patients and primary melanoma with regression areas, we could indeed identify or show by immunostaining mast cells and co-localization of CXCL10 with a high infiltration of CD3 lymphocytes, and also in the serum of patients with and without colitis, we saw a significant increase of the CXCL10. And most important, when again looking at the data from the NIH database, we do see that a high CXCL10 expression in melanoma is associated with better survival. So to sum up this project, we identified an accumulation of mast cells around cutaneous melanomas, and we could show that these mast cells can be specifically activated, leading to immune defense and tumor control. And this is mediated by the recruitment of effector T cells via the chemokine CXCL10. and the master signature as well as CXCL10 are here. So positive biomarkers for melanoma patients' survival. And this project we could then finally finish at the end of 2019. The second project I would like to present is a cooperation project with Maxime Schäfter from Gabi Multhoff's group. It's about targeting melanoma directly, or more precisely the membrane-bound heat shock protein via functionalized superparamagnetic iron oxide nanoparticles. So just briefly, Gabi did a lot of work investigating HSP70 and showed that this is frequently overexpressed in different tumor entities, and specifically on malignant cells you find this HSP70 membrane-bound, but not on healthy cells, providing an attractive target for tumor defense. And granzyme B is known to bind to this membrane-bound HSP70, get internalized, and then induce apoptosis via Cps 3. And these superparamagnetic iron oxide nanoparticles we are using, these are small crystal iron oxide that are sensitive to magnetic fields and conjugated with granzyme B. So first of all, we showed that indeed our melanoma cells we are using are expressing HSP70 on the membrane. This is shown here with a specific antibody by confocal microscopy, or here by flow cytometry with dilution of the antibody. And we could show that with using fluorescent labeled nanoparticles, that these granzyme B nanoparticles are indeed internalized via this binding to HSP70. And we could show that with the granzyme B spines, dose-dependent cell viability of the B16 melanoma cells goes down, and this is due to induction of caspase and thereby an induction of apoptosis. So going now to our mouse model, we first looked whether we can detect the granzyme B spines after injection, and we applied them intratumorally, and then looked by MR after 2 days. And as you can see, indeed, the spines retained within the tumor tissue and were detectable by MR. Then we switched to our tumor model, and now here, instead of LPS as shown before, we had now the granzyme B spines and control. And indeed, we found that these induce a tumor defense and a significantly lower, smaller tumors compared to controls. We also investigated these by immunohistochemistry with the help of Katja Schwaiger, and here the proof that we do have the particles within the tissue. We do see the granzyme B and caspase 3 induction, and we have an infiltration of CD3 positive cells. So granzyme B spines mediate here an efficient tumor defense by the induction of apoptosis. In the next step, we combined then our treatment with immune checkpoint inhibitor with anti-CTLR4, and this improved the treatment, and we really see nice tumor control now here by the treatment with granzyme B spines combined with the anti-CTLR4 checkpoint inhibitor. We also looked for the composition of tumor cells by flow cytometry, and what we noticed next to an increase in the CDC positive lymphocyte fraction, we also see a high recruitment and high increase of the NK cells. And when we looked then for the draining lymph nodes and re-stimulated them, we found that these treated mice show a strong expression of interferon gamma and TNF alpha, demonstrating that we do have here also an adaptive immune response and not only a local effect at the tumor site. So to sum up this part, so we could show that granzyme B spines target MHSP70 positive melanoma cells, remain within tumor cells and can be visualized by MRI, and that they need an effective melanoma defense with induction of apoptosis and enhanced recruitment of T cells and NK cells. We see an improved therapy by simultaneous treatment with melanoma, with granzyme B spines and checkpoint inhibitors, anti-CTLA4. So we think this combinatorial regimen of targeted nano complexes and immune checkpoint inhibitors has a high therapeutic potential that could be translated into the clinics. So I will skip this part. So finally, although I'm not personally involved, I also like to mention a cooperation project with Vasily Nitsia-Christos. He also mentioned it in this morning. So thanks to this SFB, we got 2 GERA positions, and in cooperation with Juan Aguirre, there were several studies done imaging the patient's skin, and what is shown here is a suspicious nevus that turned out after excision to be a superficial melanoma, and compared to the adjacent skin, you can see that here you have melanocytic structures also penetrating into the dermis, while here it is strictly regulated to the epidermis. And in contrast to the regulatory vessel structure, we see here in the melanoma an irregular, dense tortuous vessel structure. So this gives hope that this can be used for diagnostic in the future. So finally, I would like to thank all people involved in the project and cooperation partners, and you for attention, and yeah, thank you.
Thank you very much. Very interesting data. Any question for Dr. Kaesler? Going back to the mast cells, I mean, you showed the role in cutaneous lesions, but would it be the same in lymph nodes or metastatic lesions?
I don't know, but at least we can show that this mechanism, that the accumulation in these primary tumors is due to the mast cell accumulation. But mast cells are very very plastic cells that can change in dependency of the environment. So our idea is more that we triggered mast cells in that direction, that we can take advantage of them for therapeutic efficacy.
Thank you. If there are no further questions, thank you very much, and I will pass the microphone to Markus for the final remarks.
Thank you. Thank you very much, and after a long day and impressive presentations, thank you very much to all the presenters for the excellent lectures.